So welcome to the University of Connecticut Brain Imaging Research Center, or BERC Speaker Series. I am the host, Fumiko Haeft, Director of BERC and Professor of Psychological Sciences with secondary appointments in math, neuroscience, psychiatry, and education. Over the past 2 years, we've hosted a number of speakers, as you can see here, where most of the recordings are available on our website. We've had about 30,000 people joining our series from over 150 institutions all over the world. Thank you very much for helping us make this event successful. We've had a fantastic lineup of West Coast speakers recently, such as Dee Mobs from Caltech and Kalanit Gross-Spector from Stanford. We will continue to stay on the West Coast, including today's speaker, Russ Poldrack from Stanford. And in June, we have Terry Jernigan from UC San Diego. And in September, Marilu Gorner-Tempini from UCSF. Then we return to the East Coast in the fall with Alex Martin and Mariah Thomason in October, among others. So please stay tuned and sign up to receive information about our future talks and past recordings by emailing berk@uconn.edu. Some housekeeping items that you all know by now. You'll be muted. I usually try to unmute to clap before and after the talk, I feel like it's always nice to have this personal touch with this many people. There's going to be a major lag, and I know it's going to be a disaster, so I will not do that today. If you have questions, please enter them in the chat box. Russ is happy to answer some urgent questions that might prevent you from understanding the other parts of the talk. So if you do have urgent questions, please mark urgent and type them in the chat box, and we'll try to take it at a good breaking point. So let's get started. Today it is my greatest pleasure to introduce Professor Russ Poldrack, who most of us in our field have grown up reading many of his papers. But as you can see here, he is very young. Russ Poldrack is the Albert Ray Lang Professor in the Department of Psychology and Professor by courtesy of Computer Science at Stanford University and Director of the Stanford Center for Open and Reproducible Science. His research uses neuroimaging to understand the brain system's underlying decision-making and executive function. Russ has a bachelor's degree in psychology from Baylor University, a PhD in psychology from the University of Illinois, Urbana-Champaign, and completed his postdoctoral fellowship in psychology from Stanford University, working with John Gabriele, which makes us an academic sibling, though we've never overlapped. He has held faculty positions at Harvard, UCLA, University of Texas, Austin, and now at Stanford University. During his glorious and prolific career, he has also won numerous awards, such as the Early Career Award from the American Psychological Association, Young Investigator Award from the Organization of Human Brain Mapping, and an Innovation Award from the Social and Affective Neuroscience Society, among numerous distinguished fellowships and visiting professorships. The older folks in the room may remember some of his earlier work that used neuroimaging to pretty much investigate all cognitive functions out there. But some of the more younger people or generation may know him best for his contributions to the neuroscience community, where he has been developing neuroinformatic tools to help improve the reproducibility and transparency of neuroscience. These include OpenNeuro.org and Neurobolt.org data sharing projects and the Cognitive Atlas Ontology. I'm sure many of you use these very informative tools and have been inspired and influenced by Russ Poldrack. And I want to say here, even though he is young, as I said, he is getting a year older. Is that true today? So today is Russ Poldrack's birthday. So happy birthday to you. And when you ask questions, please congratulate him for getting a year older. So with this, please join me in welcoming Professor Russ Poldrack.
Thanks very much. It's a pleasure to be here, and thank you for the birthday wishes. Cool. So I'm gonna talk today about what I think is wrong with the way we've been doing imaging for the last couple of decades and how we might do a better job. So people have long been interested in understanding, sort of what's going on inside of our brain that gives rise to our thoughts. But it wasn't until the development, principally of fMRI in the early 1990s, actually, I guess I just missed the anniversary. It was a week or so ago of the 30th anniversary of the first fMRI, the first bold fMRI scans. This is Ken Kwong at MGH. And clearly fMRI has become a huge technique. This is a plot of just a cumulative plot of the number of abstracts in PubMed that mention fMRI. And you can see, you know, as of last year, we were at 70,000. So if you go back, like, you know, I started my postdoc at Stanford back in 1995, you know, there were on the order of 360 something papers published in 1996, up to 1996. You know, one could have, like, you know, sat down and read the entire literature with a little bit of effort at that point. Fast forward to, you know, when I moved to UCLA in 2002, there we had an order of magnitude, you know, more papers. You know, look at where we are now. You know, we're at the order of like 115 papers a week mentioning fMRI in PubMed. And so I want to ask a couple of questions. You know, we've got an order of magnitude, well over an order of magnitude more papers now than we did in 2002. Do we know, or do we really know an order of magnitude more than we did in 2002 about how mental processes are implemented in brain tissue? And maybe more importantly, will 60, or I guess that should be 70,000 more of the same kind of fMRI papers at this point answer the questions that we can't answer yet? So first, let's talk about what those questions might be. So some of the questions that we would hope that we could answer coming out of those 70,000 papers would be like, what does some particular brain area or network do? Or what brain dysfunction is associated with some particular disorder? So we would like to know, for example, what does the anterior cingulate cortex do? And, you know, there's, this is actually as of last year, there were, you know, almost 6,000 abstracts in PubMed that mention anterior cingulate and fMRI. And under some very conservative assumptions, that's well over $30 million in just for data collection, not to mention the human time that, you know, that went into to doing all that work. And so what have we learned about the anterior cingulate? Well, if you go to Neurosynth, right, which is a database that allows one to examine the association between activation locations and particular terms in abstracts, what you see is that pretty much every abstract, every term related to any sort of cognition is associated with activity in the anterior cingulate. And in fact, about a quarter of all studies in Neurosynth show activation in the anterior cingulate, suggesting that, you know, we haven't really learned very much, maybe. What about the neurobasis of psychiatric disorders? Obviously, there's been a lot of studies that have looked at case-control differences in various psychiatric disorders. And this is one meta-analysis published a couple of years ago by Emma Spruton, which compared, you know, patterns of differences across a bunch of different disorders. And this was their conclusion. Overall, there was no significant effect of diagnosis on the spatial distribution of reported case-control differences. That is, you know, in these studies, you know, sick people differed from healthy people, but they didn't seem to differ in any sort of way that was specific to any particular disorder. So how did we end up with such a lack of specificity? I'm going to argue that we've been asking the wrong questions. And so I'm going to use a thought experiment to sort of think about this. What if the phrenologist had fMRI? You know, what if somehow they'd gotten their hands on the ability to like look inside brain? So remember, phrenologists kind of felt bumps on the head and tried to map those, you know, what they thought was sort of like more or less brain tissue to different, what they call mental organs, which are basically like, you know, psychological functions that were current back in, you know, the mid 1800s, things like veneration and constructiveness and sublimity and suavity. So let's say that I'm a phrenologist and my lab studies suavity, right? We want to understand the organ of suavity. So what would we do? You know, we have fMRI, we're probably going to find some way to manipulate suavity. So I might show people pictures of suave individuals and pictures of not so suave individuals and do a comparison between those. And you would imagine I'm probably going to find a blob somewhere in the brain that differs between these sets of pictures. And I'm going to write my paper on the neural basis of suavity and publish it in a fancy journal. I kind of took this thought experiment a step further a few years ago in a paper, and I went through for each of Gall's 20, so, you know, different phrenologists had different faculty psychologies. I went through for each of Gall's 27 faculties and basically said, if I were a phrenologist and I wanted to find an imaging paper that provided evidence for the localization of my faculty of interest, could I do that? And in this case, for every one of them except for sense of cunning, I was able to find a paper that I think a phrenologist might have reasonably argued showed the localization of their faculty of interest. You might even take it a step further, right? That if they also had the internet, you might get some young phrenologist like Franz Yarkoni building a website called Neurosynth, which would basically allow you to do large-scale automated synthesis of the phrenological fMRI literature and then come up with your meta-analysis of suavity. And you might imagine that, in fact, what you would see is something like this, where you would meta-analyze all the different faculties and find that they're kind of all associated with activity in the anterior cingulate, right? Which is basically where we are right now with, sort of quote unquote multiple demand areas. So the point being that, it's not clear that the strategy that we're using is really able to answer the kind of questions that we want to answer. And I want to argue that, one way to kind of frame the strategy that much of the imaging literature is used, you can call the neural correlates strategy. That's basically where is activity in the brain correlated with some particular function of interest. So let's say that I'm interested in social cognition and I do a study and I isolate the social cognition network, right? This is actually the results of a meta-analysis for social cognition from Neurosynth. And let's say some other researcher comes along and says, hey, I'm interested in autobiographical memory. I'm going to do a study and I'm going to isolate the autobiographical memory network, right? Which happens to look a lot like the social cognition network. And then, somebody else might come along and say, hey, I'm interested in subjective value, and find that, oh, there's also some overlap between like the subjective value network and these other networks. The question is like, what leverage does this give you to predict any, right? So let's say I show you this map and say, what's going on in this person's brain? What type of task are they doing? If we were in the same room, I would have you raise your hand and tell me what you think this is. And usually people very quickly say, this is the default mode, right? This is basically somebody doing nothing except for sort of introspective thought. The point being that, that this strategy of kind of like, looking for lots of things that drive activation is not very useful for actually predicting anything. And if you haven't read this famous paper by Alan Newell, I definitely would recommend it. I think it's a real landmark paper from sort of the early days of cognitive psychology. It's titled, "You Can't Play 20 Questions with Nature and Win." And actually my initial thought experiment about like, what would 60,000 or 70,000 more papers get us is sort of stolen from this paper, where he basically said, you people are doing all these experiments that come, in this case, it was like, trying to answer dichotomies about like serial versus parallel processing or analog versus propositional reasoning, things like that. Basically saying, where is all this going to get us? And I think that, we can ask exactly the same type of question about the strategy that we've been using in neuroimaging, which you can think of as kind of a 20-question strategy, right? Does this cause the anterior cingulate to be active? Does this cause it to be active? And so on. So Yarkoni and I argued a few years ago in an annual review in psychology paper that we needed a fundamentally different strategy, which is to establish what we call selective inference. So the idea is that rather than manipulating like individual processes at once, we need to manipulate a wide range of mental processes, and then observe the associated brain activity patterns, and then try to isolate what can we predict or what can we decode from brain activity. And that allows us to provide a much more selective association between mental states or processes and brain activity. So we started trying to do this a while ago. This is a paper I published with Steve Hansen and Yara Kolchenko back in 2009, where we took data from people doing like, one of 8 different tasks. And we basically asked, can we predict which of those tasks you were doing based on your pattern of brain activity for task versus rest when we've never seen your brain before? So basically classifying based on other people's brains. And the answer is yes. We can actually do quite well. We can get, 80% or so accuracy across these different tasks. And so that suggested that this strategy actually does have legs, right? That it could actually provide us a way to start trying to understand, what particular brain systems are doing. We use some initial kind of unsupervised learning methods to try to, infer what was going on in those particular networks. I don't think it was particularly effective in that case. But in part, there's a deeper challenge, which is that, it's not tasks that we ultimately want to decode. It's cognitive processes or states or functions. Tasks are our tools that we use as psychologists to manipulate those things. But ultimately, we want to know about cognitive functions. And first, we need to know what those are. So I'm going to give you another thought experiment. Let's say you're a chemist. And there are questions that we would expect chemists to be able to answer, like what are all the enzymes? And if you're a chemist and you want to know what are all the enzymes, fortunately, there's a set of books you can go to, handily enough, called The Enzymes, which basically tell you about like every enzyme that's ever been discovered. And further, you can go to resources like the Gene Ontology, which tell you not just what are the enzymes, but where do they sit in particular metabolic processes or biological functions. So if you ask yourself, like, is there an analog of this for psychology? It's pretty clear the answer is no, right? You might hope that you can sort of go to a cognitive psychology book and say, what are all the parts of the mind? And that book might tell you, have like a big diagram or tell you like, what are all the parts of the mind? What these books generally do is they lay out the very high level parts, right? They'll talk about memory and attention and language and perception and these sorts of things. But there's no place in any of these books that I've looked at that basically lays out like, here are all the parts of the mind that we think exist and how they relate to one another. So we started a project a number of years ago to try to basically create such a resource that would characterize like, what do we think all the parts of the mind are? And then how are those parts measured by particular tasks? So this is the Cognitive Atlas. You can find it at cognitiveatlas.org. I think it's in some ways been a pretty remarkable failure in the sense that, it's gotten very little attention over the last decade, even though I think it still addresses a really, fundamental problem. And we're continuing, I'll show you, examples later, continuing to try to kind of make it useful. But let me tell you about what the Cognitive Atlas does, right? So the Cognitive Atlas basically represents two different things separately. One we call mental concepts. These are the latent psychological functions that we can't see, but that we think exist in the mind. Things like response inhibition or set shifting or response suppression. On the other hand, we have tasks that actually measure those things. And often these things get mixed together, right? People will talk about a working memory task or an attention task, right? But that's an inference about what that task actually measures that some people might buy and some people might not, right? For example, there are many people who think that the in-back task is not really a measure of working memory at all. And so we try to be very careful about distinguishing concepts and tasks and then trying to describe the relationships between particular contrasts on particular tasks and the concepts that we think they measure. And that then allows us to potentially make predictions about, for some particular task, what pattern of activation would we expect for that task, even if we've never seen it before. Now, doing this requires data from a large number of tasks. And that was part of the motivation long ago for starting to get engaged in data sharing. And that's turned out to, I think, be effective, but challenging. I'm going to show you another example of how we've done this using a data set that was collected by Maeve King, Jörn Diedrichsen, and Rich Ivory, that basically exposed individual subjects to a large number of task conditions. So this was what we call a multi-domain task battery data set, where 24 subjects underwent about 5.5 hours of functional scanning and performed 26 different tasks comprising a total of 47 unique task conditions. This is a figure from the paper. Jörn and Maeve and Rich are really interested in the cerebellum. So the initial paper sort of focused on the cerebellum. This is just kind of giving you an example of some of the types of tasks that the individuals participated in. What we've done, and this is led by John Walters, who's a grad student in my lab, and Patrick Bissett, who's a research scientist in the lab, along with Maeve and Rich. We went through and basically looked at each of the task conditions in that data set and annotated it using the Cognitive Atlas. Basically said, what do we think the the cognitive functions are that are being tapped by this particular contrast in this particular task. Usually it was just binary, like, is this, do we think working memory is present versus absent? Sometimes it was quantitative. So for example, if tasks vary in the number of response alternatives, we would annotate that quantitatively. And obviously we had to add some things to the cognitive atlas. So then what we wanted to do is take advantage of an approach that's been used pretty extensively in visual neuroscience, which is called an encoding model. And the idea is, like, if our intuition is, like, if we really understand the neural basis of cognitive processes, then we should be able to predict the activation map for any new task based only on the cognitive processes that we think are involved. And so the models that we build try to do this. So we basically, we're using region-wise rather than voxel-wise encoding models, like are usually used in the vision literature, mostly just for kind of computational tractability. So the idea is, like, in each region of the brain, for each person, we have a response, like an average Z-score across that region for each of those 47 contrasts, right? That's what the data are that we're going to fit the model to. And then the model is simply our annotation for each contrast, basically, of, you can't read these, but these are all the different, each column here is a cognitive process, like theory of mind or autobiographical recall. Each row is one of those 47 tasks. And so for each region, we fit a model that basically says, how much does this region respond uniquely to that particular cognitive function? And then we can test this using a cross-validation scheme that was first developed by Tom Mitchell back in his early work on language decoding, which we call a leave two task out procedure. So the idea is we train the model on, all but two tasks for each. We're doing this separately for each individual. Train the model for all but two tasks. And then for the left out tasks, generate, because we know for those tasks what cognitive processes we think are engaged, we generate predicted images based simply on the fitted model for each region and its response to each of those cognitive functions. And then we basically compare those predicted images for the two held out tasks to the actual images for the two held out tasks. And we just compute the correlation. And if the predicted image for task one is closer than the actual image for task one than task two, and if the predicted one for task two is closer to the actual for task two than task one, then we say we got it right. So success requires getting both of those right. So chance is around 25%. This shows how well we do when we do that. And actually we turn out to do surprisingly well, given that, part of the goal behind the Cognitive Atlas was always to kind of show how cognitive psychology is getting it wrong. And this suggests that, we actually can do pretty well at predicting what task a person's doing and predicting the activation map for a task that we've never seen before, based on a model generated from other tasks. This is each of these is a distribution across people. These are several null models, just showing that chance turns out where chance should be. So we're doing much better than chance here. We can also generalize across subjects, not quite as well, but actually pretty well. We can also look at the R squared, basically the amount of variance that we're capturing in the maps. And we can see that we're doing, I forgot to mention right here, we do best using a full ontology, but we also compared what happens if you only use more sort of perceptual motor terms versus more sort of cognitive terms. And you see that you do somewhat better with the perceptual motor terms, but you also, even if you're just using the kind of high-level cognitive terms, you can do reasonably well. And you see a similar pattern here where the cognitive terms don't do particularly well, perceptual motor terms better, and the full ontology is giving us about 20% of the variance. We're still in the process of trying to compute the noise ceiling to figure out, like, how much predictable variance is there in the maps, in terms of like the test-retest reliability, where it's pretty clear we're not quite at the noise ceiling, but we're actually not doing terrible. So ultimately, the hope is to be able to use these types of approaches to do, some sort of like meta-analytic testing of cognitive ontology. So let's say that I'm a researcher and I think that, I have these data from these 4 tasks, and I think that, these 2 tasks engage some function called inhibition, and these two engage some function called updating. But somebody else thinks that, no, there's just one function that underlies all this. And we'll call that function X, right? So now we have a way to actually test those two models, right? We can basically just ask which model provides a better prediction of brain activity on out of sample tasks, right? So we can control for the complexity of the model and test between cognitive ontologies. So we think that, by looking across many different task conditions, we can start to come up with a systematic understanding of how brain systems implement cognitive functions. Now, ultimately, right now our ontology is written in kind of natural language, many of the same terms that William James was using in 1990. But ultimately, we think that we need to cache these out in terms of a computational ontology. That's another talk, and that's sort of ongoing work. We're trying to figure out what would a computational ontology even look like. And if you're interested, I'm talking at an ontology meeting in a couple of weeks. I'm happy to send around the link to that, where I'll be saying more about that. Okay, so let's go back to one of the questions that we might hope we could answer. What's the brain dysfunction in major depressions? This is kind of a meta-analysis of meta-analyses published a few years ago by Mueller et al. In JAMA Psychiatry, where this is showing results from previous meta-analyses that had claimed to find meta-analytic significant results for case-control differences in major depression. So the idea is that each of the individual studies, I think there were like 90 something individual studies that went into all these meta-analyses. Each of those individual studies had claimed to find case-control differences. The meta-analyses claimed to find case-control differences. But when Mueller et al. Redid the meta-analyses and fixed some problems with them, they show that taking those 99 experiments, each of which had claimed to find a significant effect, they found no convergence, no significant results for emotional experiments, cognitive experiments, suggesting that there's a serious problem here. Like, you have 100 papers claiming to have found differences between depressed and healthy people, and when you put them all together, you get nothing. And in fact, this is consistent with other findings in the literature, where, for example, correlations between structural brain structure and behavior were found to fail to replicate in this early paper by Fowler-Buchel, which was the winner of the first OHBM replication award. So how did we end up with such a lack of reproducibility in the imaging literature? Well, I want to argue that we've not only been asking the wrong questions, we've been asking them in the wrong way. Many of you may be familiar with this paper by John Ioannidis from 2005, Why Most Published Research Findings Are False, which sort of heralded the reproducibility crisis that we've seen unfold over the last decade. John laid out a set of criteria, or a set of features that he thought were kind of led a field to be more likely to have false positive results. The smaller the studies, he said, the smaller the studies conducted in a field, the less likely the findings are to be true. The greater the number and lesser the selection of tested relationships, the less likely they are to be true. And the greater the flexibility in designs, definitions, outcomes, and analytical modes, the less likely they are to be true. So first, let's look at sample size. There have been a number of papers that have laid out the problems with sample size in not just in neuroscience, but kind of more generally in biomedical science and psychology. The best known one is probably this paper by Kate Button and colleagues, which looked at both structural MRI analyses and rodent studies, and saw that many neuroscience studies are woefully underpowered. A study with power of 10% or less is basically a futile study, right? Even if the effect exists, the chances of finding it are so low that you might as well just pack up and go home. And similarly, when they looked across a larger biomedical literature, Marcus Minafo and his colleagues found similar kind of poor power. Now you might say, well, that's fine. If we have poor power, as long as we control our type 1 error, then our alpha will be 0.05, and that's true. But that Ioannidis argued that may not be the quantity that we really care about, and he highlighted the relationship between power and what we call the positive predictive value. This is the kind of the flip side of the false discovery rate, right? So the positive predictive value is the probability that if you claim to find a positive result, that positive result is actually true, right? So it's, instead of like alpha quantifies across all results, posterior predictive value or positive predictive value quantifies across just the positive results. And what Ioannidis pointed out was that basically power is related to the positive predictive value, such that if you're underpowered, you're, even if, basically even when you find a positive result, it's highly likely to be a false positive. So think about it this way. Let's say you're a physicist and you're measuring something, but unbeknownst to you, your device is just giving you white noise. So let's say you do 100 studies with your device and it gives you 5%. You do that with an alpha of 0.05. So you get 5% of those studies coming out significant, but all of those are false positives, right? Your positive predictive value is 0. And then as you fix the device such that it's giving you true signal rather than noise, a higher proportion of your positive results are actually going to be true results. We also know that when you're underpowered, any positive results that you get have highly overestimated effect sizes. This is called the winner's curse. So what do effect sizes look like in fMRI? We published a paper back in 2017 in Nature Reviews Neuroscience, and then this is updating the data from that paper, showing estimated from Neurosynth sample sizes for the imaging literature, such that so like in 2019, the median study had 29 subjects per group. And that study is powered to find a single 200 voxel activation cluster that has a Cohen's d of about 0.74. And the question is that a plausible activation? Is that a plausible effect size for fMRI? The problem is you can't look at published effect sizes because they're inflated due to the winner's curse. So what we did in the 2017 paper was take the Human Connectome Project data, do a group analysis, and then basically try to find regions of interest where we were sure that there should be activation for each particular contrast. To do that, we took advantage both of the fact that the HCP group had published the regions of interest that kind of drove the selection of the task, and we used Neurosynth. So we basically conjoined these to basically say, we're going to take this region of interest, find the voxels in Neurosynth that are like, highly active for that, and then extract signal from that. And that's an independent region of interest, but nonetheless provides us with an unbiased effect size estimate for the HCP data. These are the distributions of effect sizes. And basically what we see is that, these are tasks that give pretty whopping % signal change, right? So for example, in the motor task, you're getting 4% signal change on average in the supplementary motor cortex, right? So that's about the biggest individual, like within person effect size that you can expect from fMRI. We rarely, most of the time we're looking at things that are an order of magnitude smaller than that when we do kind of interesting tasks, not like move your fingers. But that's not the effect size we care about, right? Because we're doing group analyses, we care about the distribution across people. And that's what Cohen's d looks at. These are the Cohen's d's for those activations. And you see that the biggest we ever get is about 0.7 something here. And some of them are actually substantially lower. So what this is basically telling us is that the median study published in 2019 was barely powered to find the most whopping fMRI signal you would ever expect to see. And for any sort of interesting fMRI signal, they're underpowered by probably an order of magnitude. And this gives us some leverage in understanding that the depression result from Mueller et al., right? This is plotting each individual study's sample size as a function of year of publication. I've colored the ones in with a group sample of less than 20 as having grossly insufficient N, based on this suggestion from Simmons et al., 10 years ago, basically proposing that, that it just makes no sense to do a study with less than 20 observations per cell, because a power analysis is never going to tell you that 20 subjects per cell is a reasonable thing to do for any kind of reasonable effect size for the kind of things that we study. Now, you might hope that like machine learning would help us do better, but it turns out that the same types of issues plague machine learning analyses. This is from a really nice paper by Gael Veracqua a few years ago on what he called cross-validation failure, showing how predictive accuracy values in machine learning studies also basically decline as the sample size gets bigger. So it's probably a combination of some kind of analytic flexibility, trying lots of things, and publication bias. So let's now turn to some of the other factors that Ioannidis raised regarding basically selection of relationships and flexibility of analysis. We've known for a long time that if you analyze data in a lot of different ways, you can get a wide range of results. This is from Josh Karp's 2012 paper showing he took one of the data from Open Neuro and analyzed it with almost 7,000 different pipelines and asked, like, what's the range of Z statistics that I got? And here you can see that in some brain regions, you can get a range of Z statistics on the order of 8 standard deviations, right? That's a huge difference across analyses. So we wanted to know, how does this affect analysis outcomes in the real world? And so working with a large number of people, particularly Tom Schoenberg's group in Tel Aviv, Tom Nichols and Jeanette Mumford, and then a group of economists from Caltech in Europe, we wanted to ask basically, what happens if you give a bunch of people the same data set and ask them to analyze it? So we collected a data set. Tom collected this with his group in Tel Aviv, with people doing a task that we published before, this mixed gambles test, where people see gambles like a 50-50 chance to win $28 and lose $7, and they have to just say yes or no, would you take that gamble? And 108 subjects doing one of 2 different versions, slightly different versions of the task. So we collected the data, and then we recruited analysis teams of up to 3 people each. And basically, we did it through social media and through like the neuroeconomics mailing list, because this is a task that people in that literature sort of know. We had everybody sign a non-disclosure agreement saying basically they won't tell anybody else their results until we say it's okay. And we offered them all co-authorship on the initial paper. This is why the paper ultimately had almost 200 authors on it. We gave out the raw data, and we gave them about 3 months to basically come back to us with a set of yes/no answers on 9 different hypothesis tests, each of which was of the form, is there significant activation in some particular brain area for some particular feature of the experimental design? So for example, is there significant activation in the ventromedial prefrontal cortex for like increasing size of gains in the task? And we told the groups basically analyze these data as you would in your own group. The only restriction was they had to do a whole brain analysis. So we amazingly ended up, I think we gave out the data to 84 teams, and we got back results from 70 teams, which was a surprisingly good response ratio. And in addition to their yes/no decisions, we had them give us a number of things like their whole brain statistical maps before and after thresholding. So interestingly, when we looked at their workflows, they gave us a really detailed written description based on this COVIDAS guideline. None of the 2 teams used an identical workflow. And interestingly, even with these detailed descriptions, it was often impossible to tell exactly what was done. We had to often go back to them and kind of get their code to really see what they had done. These are the main results. So what I'm showing you here is the proportion of teams that said there was a positive effect for each of the different hypotheses. And this is sorted by the average saying yes. So if everybody was exactly the same, then all these numbers would either be 0 or 1. And what we see is there's 3 hypotheses. These are all kind of like group difference hypotheses that come out to be basically, pretty much everybody agrees there's nothing there. There's one hypothesis that about 85% of the teams agree there's something there. And then there's these 5 hypotheses in the middle where 20 to 40% of the team said, yes, there's activation. Same data, right? But yet, substantial degree of disagreement between teams in the presence of activation. Another way to frame this is that if we look at the yes/no outcomes, across the 70 teams, there are 33 different patterns of yes/no outcomes. And for any particular hypothesis, there's at least 4 workflows that can give you a positive result, and many more that can give you a negative result. Now, nonetheless, if we look at the brain activity at a whole brain level, we see that there's some degree of consensus, right? But nonetheless, for example, in this map for the best contrast, this is showing a heat map of the proportion of teams who say there was activity in that voxel. The voxel with the highest agreement only had 76% of teams saying, yes, there's activation in that voxel. Interestingly, if we looked at the underlying data, this is what you see on the right here is basically a correlation map on the underlying unthresholded statistical maps that determined, whether they said, yes, there's activation or not. And you see that there's pretty substantial correlation between at least a large group of the teams. The red and green at the top shows, whether they said yes, there's activation or not. So you can see that even for teams with highly correlated maps, they could have, fairly divergent yes/no answers, suggesting that, a major part of the problem lies in the use of thresholding. Interestingly, we also saw a set of teams here, I think it was 8 teams, who were anti-correlated with the main group of teams. And when we dug into that, we basically found that they for the ones where we could actually get the details about what they had done in their model, they made model misspecification errors. They basically built the model the wrong way. And we kind of described that in the paper. So we can, at the same time, we can take the data and do sort of a meta-analysis across groups, right? So people talk a lot these days about sort of multiverse analyses, right? Of basically analyzing the data in a bunch of ways and asking what's consistent. And you see that we do find, consistent results. They don't happen to be in the regions of interest that we had kind of posited, but we do find for a number of these contrasts, except for, like these two, that there are significant results that emerge from this, sort of the meta-analysis across groups. So what this has led us to think is that we really need to focus on analysis approaches like this sort of multiverse analysis that run data through multiple plausible workflows, right? There wasn't, except for the groups that misspecified their models, there wasn't necessarily anything wrong with the way that one group had analyzed or another, right? There's sort of, reasonable divergence in how people analyze data. So what we really want to do is run the data through many analysis workflows and ask, what convergence do we get? What results are stable across analysis workflows? And obviously this requires a lot of computation. It also requires the ability to kind of sort of automatically specify statistical models. And so we've been working with a larger group developing as part of the brain imaging data structure, what we call a statistical models framework that will allow you to basically describe a complete statistical model for an fMRI data set at the both individual and group levels, basically in a file, so that you can then give that to somebody and they can rerun it and it'll run exactly the same way that it ran for you. And we have a tool called FitLens that we're developing that aims to sort of make that easy. So everything that I've told you about suggests that we've created quite a mess, right? And so we could just kind of, walk away, right? But, I think it's important we try to fix it. And so when I moved to Stanford back in 2014, Chris Gorgolewski and I started a center that we call the Center for Reproducible Neuroscience that basically aims to help improve the quality of science in neuroimaging. And so I like to think about this as a sort of a design challenge, right? We want to design a more reproducible scientific enterprise. If I was in the same room with you, I would see if anybody actually knows what this thing on the left is. Turns out it's a commercial grade paper cutter. And I would ask you, why does the commercial grade paper cutter have 2 buttons here? And you would tell me, somebody in the room would intuit that, well, that's so people don't do the thing that people are likely to do of sticking their hand to hold the paper in place and getting their fingers chopped off, right? It makes you hold both buttons down. So the idea is like the designers of this machine anticipated the errors that people would make and prevented them. And this is in general the idea behind good design, right? Is figuring out what do we want people to do and what don't we want people to do, and then affording the behaviors we want to afford and preventing the ones we don't. Another way to think about this comes from behavioral economics in terms of the context of choice architecture, right? The idea is that anytime we go into a situation where we have to make choices, like clicking buttons in SPM, there's a particular set of features that drive people toward or away from particular choices. But the most obvious and powerful one is defaults, right? By far, the default alpha threshold that people use in fMRI analysis depends on the default that's in their software, right? If you're using SPM, it's going to be 0.001. If you're using FSL, it's going to be 0.01. I don't know what it is in AFNI. But basically, there are all these ways that we can design choice architecture, which are described as nudges by Thaler and Sunstein, that allow us to basically get people to do the things we want them to do. So we focused on 3 different sort of aspects of this. One is data sharing, like providing people with tools to allow them to share data. The second is software tools to allow people to do reproducible analysis. And the underlying all this is a standard called the brain imaging data structure that I mentioned, which basically provides the glue that kind of holds everything together. So we've developed a project for data sharing called Open Neuro that basically allows people to easily upload and share data. And because it uses the BID standard, it's able to share any type of data that's supported by the BID standard, which now includes MRI, a number of different forms of electrophysiology, just recently included PET. So, so we're up over 500 data sets, up over 18,000 participants, mostly human, but also have some non-human data as well. And as I mentioned, this relies on the brain imaging data structure. This is a framework for basically organizing the data and metadata for a neuroimaging data set. And we try to make it kind of as close to what people already do in the lab, such that it's not that big of a lift to move one's data from whatever you were originally doing over to BIDs. We wanted to encourage people to use BIDs. And one of the ways that we went about doing this was like building these tools we call BIDs apps, which basically allow you to very easily apply a particular software tool to a data set if that data set is in the BID standard. So for example, if you've ever installed and try to run FreeSurfer on your own data, you know that it takes work both to install it, but also to kind of reformat your data so that FreeSurfer will know what to do with it. With the BIDs app, all you have to do is install Docker, which is the software that runs on any different platform for executing software containers. And then you just point this with our BIDs app, the FreeSurfer BIDs app, at your data set, and you give it your FreeSurfer license key. And it will basically, because the BIDs app knows about the BID structure, it basically knows how to tell FreeSurfer what to look where, and it can basically go run your data through FreeSurfer. With the added benefit that because it's using a container, all of the underlying libraries have exactly the same version as long as you use the same version of the BIDs app. So you get much higher degree of reproducibility compared to, say, running it on a machine where your sysadmin might, change the library versions. And we know that library versions can actually have an impact on results. My lab's developed a couple of different BIDs apps. One is called FMRI QC. It's always been challenging to get good quality control metrics on MRI data, or at least to get them easily. So what FMRI QC does is allow you to point your if you have your data set in BIDs format, you point the FMRI QC app at your data set, and it'll give you a bunch of kind of easily readable and usable reports on the quality of the data. Probably the most popular of our BIDs apps is this thing called FMRI Prep, which is meant to be sort of a robust and transparent pre-processing pipeline, basically that should apply to pretty much any fMRI data set. And should do kind of the best job it can with that particular data set. If it has field maps, it'll use those, and it'll sort of adapt to the data. Again, it's containerized, so it runs, reproducibly, or at least, to the degree that one can when there are random number seeds involved. And we're working on improving its reproducibility even more. And it also provides a lot of interactive reports that allow you to check the quality of particular operations very easily, right? The problem with a really powerful black box like this is that it's easy for people to just kind of run data through and not really know what's going on. So we basically try to afford them looking at reports that, at the points where we know things can go wrong easily, like in particular, the registration of structural and functional data. So we provide reports that make it easy to do that. This is basically whenever you run FMRI Prep, it unless you opt out, it sends telemetry back to us, basically telling us, did it succeed? If it didn't, what kind of errors were encountered? And so we can see that, from week to week, this is the last few weeks of last year, first few weeks of this year, we're getting anywhere from 2,500 to, over 12,000 successful FMRI prep runs. So it's getting used really substantially. I also mentioned we now have what we call a long-term support version that is meant for sort of longer term, like longitudinal projects, or projects where the data acquisition takes a long time, where you want to have basically the ability to run exactly the same pipeline on the data over the course of, say, several years. So this is something that's meant to be supported for at least, I think, 3 years going forward. So I've told you about a lot of problems. I've told you about a lot of things that we need to do to fix them. But one point that always comes up, especially from early career people, right, is that it's easy for me to say this, right? I have a fancy job. And there's a lot of challenges to actually, making this stuff happen. And I wrote a piece in Neuron a couple of years ago, an opinion piece called "The Cost of Reproducibility" that kind of addressed this, right? That, if you do all the stuff that I tell you to do, there's certain ways in which it's going to make your life much better, but it's almost certainly going to lead to fewer positive results per unit time, right? You're going to be more likely to find correct, true positive results, but you're going to have fewer positive results over time. And the problem is that, the incentives are not aligned for this, right? That hiring and tenure committees, unfortunately, still often focus on the number of publications and impact factor rather than the quality of one's research practices. So this is an issue that I and a number of others are working hard on trying to fix. There's a National Academies group on aligning incentives for open science that's about to put out a report for universities on basically how to incentivize better science. But I think this is still a fundamental problem, and we have to realize it. Okay, so just to conclude, I hope I've convinced you that we've both been asking the wrong questions and we've been asking them in the wrong way. To ask the right questions, we need to move towards selective mappings between mind and brain rather than playing 20,000 questions. To ask them in the right way, we need to implement reproducible research practices, and hopefully together these can make those next 70,000 papers answer the questions that we can't answer based on the previous 70,000. So I just want to acknowledge, all the people. This is my lab in COVID times, and acknowledge all the great people that we've worked with on all these projects and people who've funded it. And I am happy to take some questions now.
Great. Thank you very much for the excellent talk. I see a lot of clapping going on virtually, a lot going up in numbers. Does anyone have questions? Oh, Dick.
Hey, thanks, Russ. Great talk. I have a comment and a question. The comment is that I don't disagree with anything that you said with regard to the history of how fMRI has been utilized and the fact that we're gathering a lot of data now. But it seems like it's an inevitability that it would progress in that way, in the following sense. Imagine that you had gone back in 1994, the 6 of those who were doing fMRI at the time, and they went to NIH and they said, we'd like to get $100 million so that we could collect enough data to know whether the technique is going to work and to provide test-retest reliability. And of course, that just would never happen. So the luxury of the field is that over the last 30 years, enough data have been collected so that you can do open science and transparency. And this is particularly salient for me because in the modality that I'm mostly focusing on, which is FNIRS, we're not there yet, right? We don't even have the analytic tools in place that the fMRI field. Anyway, so that's just a comment. It seems kind of inevitable that we would be at this place in history with regard to fMRI. My question is, one of the reasons that you didn't discuss that I could imagine would lead to the poor alignment across individuals, even at the level of task, but certainly at the level of cognitive function, is the assumption that the anatomical templates that are used make sense. And the assumption of those templates is, obviously at a variety of different levels, cytoarchitectonics, et cetera, but the parcellation itself can be done in a different way using functional alignment techniques. And I wonder if you think functional alignment would be a better way of getting better predictability to apply to withheld samples.
Yeah, I think you're exactly right that I haven't seen the kind of knockdown demonstration yet that that functional alignment could really solve the problem in a way that, for example, a more kind of so by functional alignment, I assume you mean things like quote unquote hyper alignment, right? Actually, like aligning the maps based on sort of their pattern of response. You can also, there's work that's been done recently, for example, from the Human Connectome Project group, that's used sort of multiple aspects of data, including resting fMRI and other aspects of structural data, to align people not based on kind of like specific task activation patterns, but based on aspects of functional organization. And I think it would be really useful and interesting to ask, do those techniques when you align people based on, say, just resting, do they do better, or do they do as well as some sort of hyper alignment type of technique? Unfortunately, I think the hyper alignment tools are growing well. Unfortunately, the tools for generally doing that sort of like functional alignment from the HCP aren't publicly available yet. I don't know if they ever will be. And so those kind of shootouts haven't been done yet. But I think you're right. That's an important question to ask. And I'm hoping to hire a postdoc in the next year who has a lot of expertise in hyper alignment. So that might be one of the things that we try to go after. I think you're also right, with regard to your comment, I think you're right that it kind of had to work out this way, that any, fields always kind of stumble along in the beginning and just try to do some stuff. And, like we've seen this in genetics, right, where there were like 10 years of like all these tiny candidate gene association studies that now people realize were mostly all garbage. Now that we have like, genome-wide studies and we can see what happens when you're well powered, I think that's just often the way that science, I think you're exactly right. So this shouldn't be taken as like a deep criticism. I mean, I started doing fMRI in 1995, right? I was doing the asking 20 questions just like everybody else. So I think that's a good point to raise. So thanks.
Great, thank you. I'm just going to read someone else's comment and then move to Camille and then also to Ali. Karen Davis' comment is regarding intersubject variability. It's not just anatomy, but the strategy that different people use to achieve cognitive tasks can be vastly different. And also the theme of poor reproducibility regarding cognitive fMRI may unfairly lead people to assume this is equally a problem in some sensory motor areas. Yet there's arguably less intersubject variability in those modalities. So that's.
So I think so on the second point, regarding kind of the differences in reliability across the brain, I completely buy that. Yeah, that certainly there's a lot of reasons that sensory motor areas are likely to be less variable. I think it's an open question regarding this strategy comment, right? I mean, clearly it is the case that people, especially for complex tasks, people use different strategies. If it was really the case that drove a ton of variability, then, I mean, at the limit, fMRI wouldn't work at all, right? Because everybody would have a different pattern of activity. And, the fact that we can predict brain activity patterns across subjects almost as well as we can predict them within subjects, and what we see there is that like there turn out to like most subjects are highly predictable from other subjects, and there's a few subjects who turn out to be very different from others. And it would be interesting to know why those people differ from the others, and it may well have to do with their strategies. But for the most part, most people's patterns of activity look similar enough for us to be able to do decent prediction across these different tasks. So I think it's an open question. I think it's an open question as to kind of how broad this problem is. I agree that it's in principle a problem, and it probably is in practice a problem, at least in certain cases.
Great, thank you. Camille?
Thanks, Russ, for the great talk and for all your efforts trying to make fMRI reproducible. I have a comment to the first part or question. you didn't tell where your new perspective comes from in terms of you have now the cognitive ontology and you try to explain the data with that. But where do you get your cognitive ontology? And if it comes outside of the brain science, then obviously, why should the brain behave like a psychological theory? If it comes from the brain science itself, then it's like an iterative process. I have an idea why I do some experiment. I try to explain the results with my ideas and then see how well they explain the data and then refine it and come to a new cognitive ontology. So therefore, the opposition, what you have described, I don't see really as opposition, but more like an iterative process where we have exploration and we have explanation phase, and they sort of iteratively interact with each other. So therefore, I don't really understand why you think the cognitive ontology idea is sort of paradigm changing and not a standard process of doing good cognitive neuroscience.
Yeah, it's an interesting question. If you look back at like PET papers from the 1990s, the ones that come to mind are like early papers from Justine Sargent or like Kathy Price's stuff. People would often do these kind of detailed task decompositions, right? So like Kathy, the sine qua non of this is Kathy's cognitive conjunctions paper, where she shows basically like I have these 4 tasks, or and each of them kind of isolates a particular set of cognitive processes, and I want to find 4 tasks that only overlap in one process, right? And then I want to find regions that activate across all those 4 tasks, and that's how I'm going to decide what particular process this brain region seems to implement, right? And those are processes that came from psychological theory, right? I haven't seen one of those sorts of like task decompositions in a paper since 2000, I think, right? And I think people forgot about the fact that tasks don't just come with labels, right? That we have to, when we say like in back as a working memory task, that's a theory about, that's one person or one set of people's theory about like what's going on in that task, right? And there might be another theory about what's going on there. So the cognitive atlas was meant to kind of, help allow those kinds of decompositions, right? We have tasks that come from cognitive psychology, right? And there are theories about what go into these tasks. And when we built the cognitive atlas, we focused really only on kind of the psychological theory. We've tried to not let neuroscience really influence it because, in part because we want to figure out like where does psychological theory work and where doesn't it? Now you're right that ultimately you would hope that there would be kind of a virtuous cycle such that you could test theories, right? Test psychological theories using the brain imaging data and then allow that to feed back into psychological theory. That hasn't happened, I think, to the degree that that kind of cycle happens. I think often it's more of a vicious cycle, which is basically like a reverse inference sort of cycle, right? Where people say, I think this is a working memory task, so I'm gonna, run it. I'm gonna see activity in the dorsolateral prefrontal cortex, and I know that that part of the brain is associated with working memory, so that gives me more faith that this is a working memory task, or something vice versa. So I agree with you that strategy in principle is how we should do things. I don't think the way that it's done in the literature right now is particularly effective.
Thanks.
Great, thank you. The next question is from Ali Rahimpour. Thank you for the great talk. So you in specific highlight the lack of reproducibility in terms of spatial localization and associative brain area. Do we have the same issue for HRF in terms of bold response across the time, bold temporal changes? Also, have you studied this issue in ERP-EEG, direct neurophysiological activity as well?
Yeah, those are both great questions. I think we don't know for HRF is the short answer, I think. And the problem is that in order to characterize the nature of the HRF, in order to characterize the hemodynamic features of the HRF, you have to know how to drive neural activity in a particular way, right? The reason that we know this, say, for visual cortex is because we know from electrophysiology, for example, the function that relates contrast to neuronal activity. And then we can infer from that kind of what's the, how linear is the relationship, or, how variable is the relationship between neural activity in V1 and the hemodynamic response, right? For most of the brain, we don't know how to drive neuronal activity directly in a particular way that we could then use to assess, so for example, there were, early on, there were these debates about these really long drawn out responses in ventromedial prefrontal cortex on memory tasks, right? And it just wasn't known, is that because this part of the brain just has a weird hemodynamic response, or is it because there's long lasting changes in neuronal activity? The problem is that's an area where we don't know how to drive punctate differential levels of neuronal activity. So I think we just can't answer the question for much of the brain. I haven't looked at these issues in ERP-EEG. There's a great paper by Nick Gastelum and Steve Luck from a few years ago that basically tried to argue that all the same issues around at least around analytic flexibility are present in ERP analysis, just as they are in fMRI. So I'd recommend having a look at that paper.
Great. Carlos?
Well, first of all, thank you for your great talk and clear slides. I understand what people were talking about, the contrast and subject anatomic differences and the brain templates. I was thinking when you think that when we measure that fMRI response, we have this around between 6, 8, maybe 10% of changes which we use to measure fMRI. I believe all these differences somehow we should take care when we make statistical analysis. For example, I remember a few years ago we used to mostly use family voice error, and now I can see several studies using false discovery radio. So I don't know if you have seen about these kind of differences in the statistics. I mean, I believe this could make a big difference. Thank you for your program.
Thanks. Yeah, no, I think you're right that there's a number of different ways that one can analyze data, and the real challenge comes about when people try lots of those things and take the one that gives them the best result, right? So this interesting thing happened about a decade ago. My colleagues and I were noticing that a number of researchers would publish results where they analyze all their data with FSL, but then in the end they use this AFNI tool, 3D ClustSim, to get their p-values, right? And that's weird, right? Because FSL comes with tools to give you your corrected p-values. Why would you go use another tool? Well, it turned out that AFNI tool was systematically biased towards giving you more liberal results for reasons that came out in this 2016 cluster failure paper. And so this suggested to us, at least, that people were basically p-hacking in the open, where they were basically saying, well, my tool gives me a way that I know should control either false discovery rate or family-wise error, but there's this other thing that says it's controlling family-wise error and it gives me more activation, so I'm going to go use that one, right? Turned out that it was wrong, right? So that's the big concern that I have. I think that that as long as somebody, as long as they're consistent in the way they do it, and preferably if they pre-register it and say what they're going to do, then that at least takes away the flexibility is the part that I worry about. I think as long as you're controlling something, that's a great first step. And I think, controlling FDR or controlling family-wise error are both kind of reasonable things to do, assuming that the tool is working properly.
Great. Maxim?
Hi, well, great talk, very stimulating, very interesting. So I have a kind of basic question of what do you think about basic limitation of the method? Because, well, both signals certainly is kind of far cry of neuronal activity, right? And certainly and obviously, the technique is very well very well differentiate between activation of motor cortex or visual cortex or prefrontal cortex in very specific tasks. But what do you think is a limitation, intrinsic limitation, for disentangling what exactly the brain is doing? I mean, again, you can certainly tell the difference between motor cortex and visual cortex, and in the motor cortex, probably you can tell difference between moving fingers and moving the whole hand, right? But in the visual system, the technique definitely cannot tell whether you are looking at this face or that face, unless the faces are have some very different emotional load. So what do you think about this overall limitations?
I mean, in some sense, so I agree. Yeah, clearly there are limitations of fMRI that have to do with, various aspects of resolution. In part, it depends on exactly what you mean by what the brain is exactly doing, right? Because I think what we've, one of the things that fMRI has really helped us learn is that there are multiple reasonable levels at which to look at brain function. It's not as if only looking at I think, 20 years ago, a lot of the hardcore, monkey electrophysiology people would have said, well, if you're not studying spikes, then you're not really studying what the brain is exactly doing, right? And clearly spikes are part of what the brain does, but there's a lot of other stuff that we now know that the brain does, right? Around oscillations, around, more global field potentials, all that sort of stuff. And I think that, what fMRI has shown us is that there's a level of organization, there's kind of a mesoscale level of organization, or multiple levels actually, right? There's this level at, what people call kind of resting networks, right? Somewhere between say 7 and 15, 20 different sets of regions that move together. There's this finer grain organization of regional stuff, right? That has come out of all the regional parcellation stuff that Steve Peterson and Nico Dosenbach's groups and others have done, that are probably on the order of a centimeter or two, and those seem I think it's been nicely established now that those are real organizational features in the brain, right? Even though they come from resting fMRI, they map well onto task activation. There's some electrophysiology data suggesting that they do reflect, differences in physiological responses. They're not just some artifact. So I think it's not as if fMRI is not measuring exactly what the brain's doing, right? It's measuring exactly what the brain's doing at a particular level. It's not able to measure other levels. And I completely agree that, particularly with regard to temporal information, clearly, we're seeing, when we see an fMRI response, we're not seeing the first pass of neuronal activity through the network, right? We're seeing an average of that stuff bouncing back and forth across the whole brain for, a few seconds. And that's clearly, could mislead us into thinking, certain things are going on. And so I think that, we always have to be, very clear about the limitations. I'm excited about, the fact that data sharing for EEG and MEG is increasing now that there's BID standards for those. Open Neuro is getting an increasing amount of data. And so we might be able to start doing the same kinds of things with those measures that we can with fMRI to help us get a kind of a different picture.
Yeah. Thank you.
I'm going to go and take Yin Wang's, a new person's question, and then I'll do Karen, and then I'll do Camille. And this will be the last call. I know we have several more minutes left. So Yin Wang's question is, thanks a lot for the excellent talk. Just a thought, the individual differences in imaging data might not be actual noise, especially for higher order cognition. Can you comment on whether your pipeline can look into individual variances in functional imaging data, which might be unique individual characteristics?
I think that's a great point. And, I first became convinced in this growing out of the myconnectome project, where I scanned myself a ton, and we saw that there were these weird network features in my brain. And initially we didn't know if I was just weird or, if they were a common feature. And now we know from all of the work from Nico Dosenbach's group in the Midnight Scan Club project that that they really are like every individual has like, you know, these little variant functional organizations in their brains, little spots that, you know, should be part of the default mode network, but are actually part of the salience network, for example. And so in my lab, you know, we've really moved away from doing the kind of imaging studies we used to do where we kind of, you know, take a bunch of individuals and scan each one for an hour and try to do something with that. We're scanning individuals much more densely. So, you know, for example, we have a study now where people come in, I think 12 times over 6 months, and we, you know, scan them so in a way that allows us to actually pull out, you know, ultimately like model individual brain networks, so we can address exactly this question. Because I think you're right, a lot of it is not noise. If we have enough data, we can actually see, you know, reliable systematic individual differences.
Great. Related to this, Karen Davis asked a question. So she said, thanks, Russ, for getting us all back to thinking about the big issues. So important to keep in mind. So what do you think about N of 1 studies? In the clinical world, the single patient finding can teach us a lot.
No, I think that's exactly right. You know, it's challenging to know how to interpret an N of 1 study, right? And that's, you know, going back to exactly what I just said about my connectome, right? Where we didn't know initially whether, you know, it was just me who was weird or whether it generalized. I think we, you know, so it takes more than just one often. And obviously, you know, HM is a great example, right? HM, you know, provided kind of a proof of concept, but we, you know, it took a lot of studies of a lot more people to finally kind of, you know, believe that, you know, what that case study told us. But I think that, you know, the nice thing about a case study is it provides you with like really solid evidence regarding one person in a way that, you know, you can't often, you know, the evidence is often much less certain when you're looking at a bunch of people and you only, you know, collect a little bit of data on each person.
Great. So last question, Camille.
Thanks again for picking me second time. So Russ, can you comment on the word reproducibility? And the reason why I'm asking is Eklund made sort of the paper, famous paper saying all 40,000 fMRI papers are wrong. And this was what he literally wrote. And then he modified after criticism saying when they are weakly significant. But most good fMRI scientists knew you can anyway not trust weakly significant data. So it was then a trivial statement. And the reason why I'm asking what the word reproducibility means is, doesn't it depend on your statement, what you are extracting from the data? If I would, for example, say this stimulus evokes something in the brain, then all analysis pipelines will agree with that. And if I then say, make another statement with the same data, with the same different analysis tools and say, this voxel in that exact location is activated, and somebody else finds the neighboring voxel activated, then it would be not reproducible. So instead of, perhaps it's not reproducibility crisis, it's an overstatement crisis, that the people do actually overstate and overinterpret their findings instead of being careful and saying this is what we have found and this is how we interpret and these are the limitations. So instead of, I'm thinking whether it's really the reproducibility which is the problem, but that people try to say we have now solved 2000 years of philosophy discussion on freedom of will with doing this experiment with p equals 0.049. So I think it's a more, so that was my question.
Yeah. So I think that's a great point. I mean, and lots of, you know, reproducibility means lots of different things to lots of different people. There's a recent paper in BBS by Tagliarconi about what he called the generalizability crisis, which kind of gets at exactly what you're saying, right? Because in part, right, I don't care if this exact voxel reproduces, right? What I care is that there's a scientifically relevant statement that I can generalize from my finding, right? So it's not like this voxel at -3/16/24 is active when I see faces, but there is a region in the fusiform gyrus that is, you know, systematically more active when people view faces than other things, right? And that's something that you can go generalize to another person. And obviously you have to define, how do you find, you know, what is the fusiform gyrus? How do you define that? But I agree that, you know, that there's a lot of layers, right? We've been focusing on kind of the the more boring layers of like, making sure that when you analyze the data this twice, you get the same answer, right? Or that if two people analyze the data slightly differently, that they come up with the same answer. But same answer is a really challenging thing to define, right? And that's exactly what you're getting at. And I think there was some work that I think Stefano Anzalotti was doing on this, and I didn't ever see it published. I was trying to get at like, what does replication mean in neuroimaging? And I think that there's actually still interesting work to be done there. I think that hasn't been kind of fully fleshed out, but it's really important to keep in mind.
All right. Thank you very much for staying on for such a long period of time, Russ. I think it was a very exciting talk. Especially thank you for joining on your birthday. I didn't hear anyone wishing him happy birthday, but I'm sure everyone was thinking so as they were listening to your talk. So thank you very much. And I hope you have a wonderful rest of your day. And thank you everyone for joining as well.
Yeah, thanks. See you all.
Bye. Thank you.